Do you love carbs? I freaking love carbs. I could live off of bread if it was physically possible. Fortunately, there's not enough vitamins and nutrients or protein in there, but if I could, I would eat just bread. Okay, but let's talk about the biochemistry of carbohydrates and what's going on here, okay? So remember our 4 categories of biological molecules: carbohydrates, lipids, proteins, and nucleic acids. Okay, each one has a different structure, different function, and different role that it plays in cells. So here we're going to dive into carbohydrates. Okay, here's our big picture, where we're going today. Carbohydrates, they're monomers, monosaccharides. Their main purpose is easy access to energy, quick access to energy for cells. How they're arranged, how they're built, how they're deconstructed, dehydration, synthesis, and hydrolysis. And major types of examples that you should know from the reading: glucose, lactose, sucrose, glycogen, starch, and I'd throw cellulose in there as well. Okay, so monosaccharides, these are the sugars. These are kind of simple carbohydrates, you could say. Mono meaning one, and saccharide meaning sugar in Greek. So these molecules, let's take a look at them, right? Remember, polar bonds are hydrophilic. Anything that is a polar bond is a hydrophilic molecule, or has hydrophilic capacities. So remember oxygen, carbon and oxygen is a polar bond, oxygen and hydrogen is a polar bond, right? And look at all these oxygens in here. Look at all these hydroxyl groups, okay? So because of this polarity, because oxygen is really greedy with its shared bonds, carbohydrates are very hydrophilic. They will readily dissolve in water, okay? Because this negative oxygen will readily bond or make a hydrogen bond with water molecules, with the positive ends of the water molecules, okay? And when they're dissolved in water, they tend to take on a ring shape. So we'll see often carbohydrates kind of drawn out in these 2 different ways. These are the same molecules, okay? But usually when water is present, they take on this ring shape like this. Okay, so same molecule, just slightly different shape. We're just kind of taking this side over here and kind of reattaching it. Their main purpose, monosaccharides and disaccharides as well. So if we connect 2 of these together, which we'll talk about in a second, how that happens, but if we connect 2 of these together, like glucose and fructose, monosaccharides and disaccharides, their main function is easy access to energy, easy access to a molecule that's really useful in building ATP, adenosine triphosphate, the energy for cells. Our cells, animal cells especially, are really, really good, very efficient at breaking down monosaccharides and disaccharides into ATP, which we'll talk about later when we get to cellular respiration. But that's their main function, especially for mono and disaccharides. Their main function is easy access to make ATP. So most carbohydrates, monosaccharides included, have the same basic formula, CH2O. Okay, so any variation of this, we could have C6H12O6, that's glucose. Okay, so any multiple of this number gives us a carbohydrate. So carbon and water is how they get their name, right? Carbon, H2O is water, carbohydrate. Okay, so we're taking a carbon-based molecule and we're hydrating it basically, quote unquote, with water, in a sense. Okay, so ring shape in water. This is the exact same molecule though, just an isomer of it, right? And they are hydrophilic, and their main purpose is easy access to make ATP, a molecule that's really easy for cells to make ATP out of. This is what it looks like when glucose gets folded up into a ring, right? We are basically breaking apart this bond up here and then bringing it over to make a bond here. Okay, so it's folding up. But the direction in which it folds up, the direction in which this OH ends up, this hydroxyl group, if it's facing down, we say it's alpha glucose. If it's facing up, we say it's beta glucose, right? And just a reminder, when we're looking at these types of diagrams, each point right here See how there's no atomic symbol written there? There's no H or N or anything. When we're looking at biological molecules, if there's a point where two lines connect and there's no letter written there, we can imply that there's a carbon. Right, so here's a carbon here, there's a carbon here, and here, and here, and here. We're just not writing them in because there's just so many of them. Right, so we still have 1, 2, 3, 4, 5, 6 carbons, just like we have here, just looking at it slightly differently. Okay, so depending on which way it folds up, we can have either alpha or beta glucose. And this is important because even that slight difference in where this hydroxide group is pointing makes it so our cells, the enzymes in our cells, might not be able to interact with that molecule. So like beta glucose, this one right here, is the monosaccharide that's used to build the molecule cellulose, which our cells cannot handle. Our cells are like, what is this? This isn't the right shape for my enzymes to do anything with, so it has no value to us nutritionally. Alpha glucose, though, we love the alpha glucose. It's part of starch, it's part of glycogen. Starch is found in plants, glycogen is found in animals. Talk about that in a minute. But alpha glucose, our cells are like, yep, I know what this is, I can handle it, I can break it down, and we can get energy out of it. Okay, so just that slight difference in where that hydroxide group is facing on that first carbon makes it so our cells can or cannot interact with it. All right, so let's connect together some of these monosaccharides to make a poly or a disaccharide. Okay, here we're making the disaccharide sucrose. So you probably have sucrose in home. You probably have eaten sucrose today if you've eaten anything at all. Most foods here in the States have sucrose added to them. But what's going on? Okay, so sucrose is when we take the monomer, the monosaccharide glucose, and the monosaccharide fructose, and we connect them together. Most biological molecules will be built through this process that we call dehydration synthesis. So dehydration meaning to remove water, and synthesis meaning to build. So basically what's happening is here's our alpha glucose here. Here's our fructose molecule, right? So these are two separate monosaccharides, but we can connect them together. We can build a bond. So basically, we're going to take this hydroxide group right here, and this hydrogen from this hydroxide group, those are going to connect together to make water. And now this carbon right here will bond to this oxygen. We call that a glycosidic linkage, but I'm not too worried about that. I'm more worried that you understand the idea that we remove water. So water is a product during the dehydration synthesis reaction. And that's how we connect together monomers to make a polymer. In this instance, we are connecting glucose and fructose to make the molecule sucrose, which is just normal table sugar. Like if you have a bag of white sugar in your pantry, that's a bag of sucrose, basically. Okay. And we can keep doing this. We can attach some more here, keep building this on to make longer molecules. Okay. But the important part here is that dehydration synthesis, we remove water to build larger molecules. To connect together monomers to build polymers is dehydration synthesis. So you should be able to draw this out and recognize this reaction and describe this reaction. All right, so good sucrose. I have a really bad sweet tooth. And this week just brings it out in me. It's terrible. Okay, but there are more carbohydrates than that, right? We have our simple sugars like sucrose, glucose, fructose. Lactose is the sugar that's in milk. It's built the same way that we just talked about with sucrose, right? But if we connect together more than two monosaccharides, so more than two, we start to call it a polysaccharide. Poly just meaning many. We're taking many of those monomers, those monosaccharides, and we're connecting them together. Usually into very, very, very long chains, hundreds, thousands of monosaccharides long. Some might have branching, some might not, right? So these polysaccharides have a slightly different function than the monosaccharides. They're still often used for energy, but it's a form of energy storage. Okay, so it's a way to form energy so that way we can access it, but not quite as readily accessible as a monosaccharide. We also see that these polysaccharides are good for structural integrity, especially in plants and fungi and insects. They use a type of polysaccharide that's very rigid and gives them structural support to either their exoskeleton if they're an insect or to their cell wall if they're a plant. We don't use them, humans don't really use them for structural integrity because our cells don't have cell walls. We do use polysaccharides often for cell markers to indicate what type of cell each cell is. Is this a liver cell? Is this an eyeball cell? Is this a stomach cell? Is this a red blood cell, white blood cell, right? All those differentiations between our cells need to be able to know who they're talking to, who they're connected with. Okay, and a lot of that marking is done, identification is done using polysaccharides. So common types that we should know: glycogen is a very common polysaccharide in animal cells, and starch. We have a couple different types of starch. These are typically found in plant cells. Okay, main types are going to be amylose and cellulose. Okay, and both of those are produced by plants, which we then use. All the carbs. Give me all the carbs. So, long chains of glucose are really what most of these polysaccharides are. At least these are really important ones that we're talking about. But they're going to vary in the number that are in these glucose chains and the type of bond that's there, and if it's an alpha or beta glucose. Okay, so like let's take for instance in a potato. Potatoes are a starchy vegetable, right? That starch that's in potatoes, that's a really great source of calories for us, is from amylose. So see, we have lots and lots and lots of glucose connected together, and there are a few branches off of this molecule, but not many. Mostly amylose is just really long chains of thousands of glucose molecules connected together. And these tend to be, in amylose, these tend to be alpha glucose, the kinds that our cells can use and interact with. Okay. In glycogen is slightly different. So look at the difference between this glycogen molecule and this amylose molecule, right? We see there's still really long chains of alpha glucose, but we see a lot more branching. So we're connecting together multiple kind of lines and strings of those polymers together. So glycogen is the molecule that's very commonly found in our muscle cells and in our liver. It's a way for us to store glucose so that way we can get to it pretty easily later if we need to, right? So like if you haven't eaten in a while, your blood sugar starts to drop, our body can then break apart these glucose molecules and re-release them into our bloodstream so that way our cells have a really easy to use form of energy. Okay, so glycogen is a way to store glucose for later usage without having to convert it to fat. Versus cellulose, look how different cellulose looks. Also, what do we notice about the bonds here? The orientation of these glucose molecules. These are different. These tend to be beta glucoses, right? The hydroxide group is on the other side. So cellulose, we don't have cellulose in our cells. It's only found in plant cells. And it's really, really long kind of cables of that beta glucose connected together, and you can tell because based on the orientation of those oxygen molecules. Look, oxygen here is all facing down, oxygen's here all facing down, here it's alternating. Okay, that's how we can tell it's different. So our cells can't do anything with cellulose. We can't break it apart to get energy out of it, but it's really great for plant cells because these long cables can then be connected together through hydrogen bonds and create these fibers that are really great for providing structural support. So they're not useful for us as far as getting calories out of, energy out of, but they provide fiber roughage, if you would, in our diet to keep everything moving. So these, our cells, can break down glycogen and can break down starch. We can cleave off these glucose. We can break that bond that was built during dehydration synthesis through what's called hydrolysis reaction. So hydrolysis is the opposite of dehydration synthesis. It's how we take a polysaccharide or a disaccharide and we break it back down into its monosaccharides. Because those monosaccharides, that's the form of carbohydrates that our cells are really good at using to make energy out of. So it's the same, basically same reaction as dehydration synthesis, but in reverse. So hydro meaning water and lys meaning to break. Okay, so we use water to break apart this bond between our monosaccharides. So water in this sense is a reactant, not a product. Water goes into this reaction and is used up and basically reattached, separated, so that way we can separate those monosaccharides. So hydrolysis is how we turn a larger molecule, a polymer, into its monomers. And we'll see this with other biological molecules as well. Hydrolysis is the main reaction for how we break down those polymers into their monomers. And then these monomers, our cells are like, cool, glucose, fructose, those are easy to get ATP out of or to make ATP from, so these are good to go. But notice, there's something here that wasn't there before. This molecule right here, the sucrase, this is not a reactant, it's not a product, it's a catalyst, and it's an enzyme. And we'll talk about enzymes later, but enzymes are catalysts that speed up reactions. So in our cells, we have sucrase enzymes that break apart sucrose. We also have amylase enzymes that break down the polymer amylose, right? That speed up this hydrolysis reaction. So that way our cells can access and use those monosaccharides for energy. Okay, a big takeaway from carbohydrates. Let's break it all down again. They're very hydrophilic, right? Because they got all those oxygens on them. Oxygen is very polar when it bonds to carbon or hydrogen. Monosaccharides are their monomers, right? They're building blocks, they're single pieces that we connect together are the monosaccharides. And those monosaccharides are most often used for ATP production. Okay, we can use those monosaccharides to make ATP in our cells through the process of cellular respiration. We connect together monosaccharides and we get a polysaccharide. So the polymer of carbohydrates is polysaccharides. And how do we build them? What process builds them up? Dehydration synthesis. So we remove water to make a big molecule to connect together the monomers. And polysaccharides are often used for structural support in the sense of cellulose, or energy storage. Again, dehydration synthesis to build polymers, connect together monomers to build those polysaccharides. And hydrolysis is the opposite. Hydrolysis breaks down those polymers, so that way we can get back to the monomers and then use those monomers to either be converted into another molecule, or most often those monosaccharides are used to make ATP. Alright, carbs are not bad. Everything in moderation, but I promise you carbs are not bad. Our bodies need carbs. So go enjoy a few carbs, just don't go too crazy.